Good afternoon, CM students. So for today, I will be discussing your CSF analysis. Your cerebrospinal fluid class, it was actually discovered by Cotugno, and it is a major body fluid. Now, your CSF, it provides a physiologic system to supply nutrients to the nervous tissue, as well as it removes metabolic wastes, and it produces a mechanical barrier to cushion the brain and the spinal cord against trauma. Now, some important functions of the CSF includes it provides nutrient, or rather it serves as a nutrient medium of the CNS, as well as it acts, or rather it functions to provide physical support and protection against sudden changes in acute venous and arterial blood pressure or impact pressure. And aside from this one, it serves as an excretory channel for metabolic products of the CNS. And then lastly, it maintains a CNS ionic homeostasis. Now, indications for performing lumbar puncture and CSF examination includes suspicions of encephalitis and meningitis, as well as multiple sclerosis, neurosyphilis also, and subarachnoid hemorrhage. Now, this procedure is usually performed also on patients with unexplained seizures, and as well as those patients who have fever of unknown origin. Now, let us talk about the formation and physiology of the CSF. Okay, as we all know, the brain and the spinal cord, they are lined by the meninges, okay, which consist of the three layers, namely your dura mater, the arachnoid, and the pia mater. Now, the outer layer, which is the dura mater, it lines the skull and the vertebral canal. Now, the arachnoid naman, it is a filamentous. Sometimes we describe it as, spider-like structure, serving now as the inner membrane. And then the pia mater naman, it is a thin membrane lining the surfaces of the brain and the spinal cord. Now, for the formation of the CSF is produced in the choroid plexuses. It is produced in the choroid plexuses of the two lumbar ventricles and the third and fourth ventricles. Now, in adults, approximately 20 ml of fluid is produced per hour. Again, among the adults, approximately 20 ml of fluid is produced every hour. Now, this fluid, yung CSF natin, okay, it flows through the what we call the subarachnoid spaces located between the arachnoid and the pia mater. Okay, so kung imagine natin yan, nandito yung pia mater natin, which is the inner membrane, and then this is the arachnoid, and then and then here is the subarachnoid spaces. Nandyan. Okay, parang baliktad. Okay? Ayan. Now this, again, the fluid, it flows through the subarachnoid spaces located between the arachnoid and the pia mater. Now, to maintain a volume of 90 to 150 ml in adults and 10 to 60 ml in neonates, the circulating fluid is reabsorbed back into the blood capillaries located in the arachnoid villi. Again, ulitin ko, yung fluid na yan na dumadaloy sa subarachnoid space, okay, it is being reabsorbed back here in the capillaries of the arachnoid granulations or villi, as we call it. Okay? It is being reabsorbed at a rate equal to its production. Again, class, huwag niyong kakalimutan, it is being reabsorbed back here in the arachnoid villi at a rate equal to its production. Now, the cells of these arachnoid granulations, it act as one-way valves that respond to pressure within the CNS and it prevents reflux of the fluid. Now, in terms of CSF distribution, your CSF, it is distributed in the ventricular system as well as in the subarachnoid spaces of the brain and the spinal cord. Now, in the choroid plexuses, the endothelial cells have actually very tight-fitting junctures, okay, that or whatever, okay, that prevents the passage of many molecules. Now, this tight-fitting structure of the endothelial cells in the choroid plexuses is termed as the blood-brain barrier. Ito yon actually. Now, there are two morphologically distinct components of blood-brain barrier, namely as the capillary endothelium and the choroid plexuses. Now, the functions of the blood-brain barrier, the functions of which it includes protection. Okay, protection, tawag dito, or rather, it protects the brain from foreign organisms in the blood. Okay, as well as, tawag dito, wait lang class. Okay. Ayun, ulit. Okay, again class, maintaining the integrity of the blood-brain barrier, it is essential to it is essential to protect the brain from chemicals as well as other substances circulating in the blood that could harm the brain tissue. Now, in contrast, the junctures also prevent the passage of helpful substances including antibodies and medication. Then aside from this one, the functions of blood-brain barrier also, it shields the brain from hormones and neurotransmitters from the rest of the body. And it also maintains homeostasis for the brain. Now, let us proceed to cerebrospinal fluid analysis proper. Again, indications for CSF analysis includes the presence of meningeal irritation, conditions involving the CNS, kung meron ding increased intracranial pressure, what else? For the purpose of drainage. When we say drainage, pag merong subdural hematoma. And then for the introduction also of X-ray contrast media as well as introduction of drugs. Okay, so yung mga procedures na yan or conditions, yan ang mga indications natin for performing CSF examinations. Now, for the specimen collection and handling, the methods of collection includes lumbar tap, pwede rin cisternal puncture. I think it is in your handout naman. Okay, cisternal puncture, lateral cervical puncture, and even ventricular puncture. Now, sa lahat ng ito class, CSF is routinely collected by lumbar puncture. Yan ang actually pinaka-method of choice when it comes to collection. Okay? Now, lumbar puncture, it is done between third, fourth, or fifth lumbar vertebrae. Okay? So, ito yung mga tawag dito, mga site na pwede tayo mag-collect ng CSF. Now, although this procedure is not complicated naman, it does require certain precautions, including measurement of the intracranial pressure and careful technique to prevent the introduction of introduction of infection or the damaging of the neural tissues. Now, the CSF pressure, it is measured using a graduated manometer attached to the syringe. Okay? Again, CSF pressure, it is being measured using a graduated manometer attached to the syringe. Now, the normal intracranial pressure among the infants, it is usually 10 to 100 millimeters of water. And then, for the adults naman, For the adults, it is usually 90 to 180 millimeters of water. Now, we have this what we call test. We have your Queckenstead test. Okay, this method class, it is used to confirm subarachnoid block. Again, your Queckenstead test, it is used to confirm subarachnoid block. Now, yung procedure na yan, class, ang principle niyan, normally daw, if both jugular veins are manually compressed, the CSF pressure will rise rapidly to over 300 millimeter. Okay? And then, normally din, class, it should rapidly return to normal when compression ceases. Okay, so that is now the principle of your Queckenstead test. Indication to it is used for confirming subarachnoid block. Now, typically, class, 10 to 20 ml is collected. Okay, when it comes to collection of specimen, 10 to 20 ml is enough na yan for collection of CSF. Okay, it is collected into 3 to 4 sterile tubes that are numbered sequentially. Okay? So, i-label mo yan, tube 1, tube 2, tube 3, tube 4, and ayun. Okay? Now, each of this test tube, they have separate uses. Okay? May mga designated area yan sa laboratory. Now, tube 1 class, it is used for chemical and serologic testing. It is used for chemical and serologic testing. Because this test class, they are least affected by blood or bacteria introduced as a result of the top procedure. Okay, so hindi sila masyadong affected by blood or bacteria being introduced as a result of the top procedure. And then we have tube 2. Your test tube number 2 naman, it is usually designated for microbiology laboratory, because it is the least likely to contain cells introduced by the spinal tap procedure. Okay? And then your tube 3 naman, it is used for cell count. Usually, mapupunta siya sa hematology for cell count. Okay, now tube 3, because it is the least likely to contain cells introduced by the spinal tap procedure. Now, a fourth tube class may be drawn for the microbiology laboratory to provide better exclusion of skin contamination, or pwede rin ibigay mo sa zero kapag kinulang ng sample. Okay, so dalawang choice natin kasi excess tube lang naman yan. I mean, excess sample for test tube 4. So, pwede siyang mapunta kay microbio kasi it provides better exclusion of skin contamination, or pwede siyang mapunta kay serology. Now, take note also, class, na pag sa laboratory, for example, in the future, kung maka-encounter man kayo ng receiving these kinds of sample, let's say CSF, kapag may excess fluid, it should not be discarded. Immediately. Okay? But rather, it should be frozen until there is no further use for it. Okay? So, itago nyo lang kapag may sumobrang sample kasi mahirap nga namang i-collect yan. Okay? So, for example, may repeat testing, ganyan. So, pwede natin gamitin yung excess sample natin. Now, for the storage of these samples, may kanya-kanya din silang proper storage. O, tama. Okay? Now, ideally, that these tests are performed on a stat basis. Dapat stat yan lagi. However, if this is not possible, specimens are maintained in the following manner. Okay, so yung tube 1 natin, yung for chemistry and serology, the tubes should be frozen. It should be frozen. While your tube 2 naman, your microbiology tubes should remain at room temperature. It should remain at room temperature, class, lalo na kung suspected cases yan ng meningitis. Remember, your let's say your Neisseria meningitidis, they are very cold sensitive organisms. Kaya kailangan ilagay mo lang siya sa room temperature. Although yung ibang laboratory class, nilalagay nila sa 37 degrees Celsius before performing plating, bago nila ilagay sa culture media. But sa references, we have to follow this one. Okay? It should be stored at room temperature. And then lastly, your tube 3 naman, your hematology, they should be placed in a refrigerated temperature. Now let us go over with the physical examination of your CSF fluid. Now, a normal CSF class, a normal CSF specimen, it should appear as clear, colorless, and less viscous dapat. But pathologically, a cloudy or sometimes turbid or milky specimen can be the result of an increased protein as well as increased lipid. Okay? So again, a cloudy specimen or sometimes turbid or even milky specimen can be the result of an increased protein or lipid concentration. But this one class, this type of specimen, it may also be indicative of infection with the cloudiness being caused by the presence of WBCs. Okay? So kung cloudy yung specimen natin, pwede tayong mag-suspect ng merong infection. Okay? Due to the presence of WBCs as well as microorganisms, okay, mga bacteria, ganyan. And then, pwede naman class, physiologic causes of a cloudy specimen is due to radiographic contrast media, okay, or radiographic contrast materials, as well as aspirated fats. Okay, yan yung mga physiologic causes natin ng a cloudy specimen. Okay. Now, yung cloudy specimen nating class, yung mga concentrations ng ating WBCs as well as RBC and protein, yung mga concentrations nila, please refer na lang to your handout. And then next, we have your bloody CSF specimen. Now, a bloody CSF specimen could be an indication of a traumatic tap or intracranial bleeding. Okay? Tapos, yung visible bloody CSF sample natin class, it is usually caused by a more than 6,000 RBCs per microliter. Now, however class, lagi niyong tatandaan that a clear CSF sample, it does not necessarily rule out intracranial or intracerebral bleeding because an RBC count of less than 360 per cubic millimeter, okay, it will not give a visible bloody specimen. Okay, do not forget this one. Now, how do we differentiate your, or paano natin malalaman kung yung bloody CSF sample natin is due to traumatic tap or due to intracranial bleeding talaga, which is a more significant finding. How will we differentiate the two? Okay? So, tanggalin lang natin to. Let us differentiate your traumatic tap and a true bleeding. Okay, let's say hemorrhage. Okay. Now, in terms of their distribution class, okay, nasa handout nyo yan, i-fill out nyo na lang. Okay, in terms of their distribution, a traumatic tap, kapag yung kanilang distribution is uneven, traumatic tap yan. Okay, ano yung sinasabi kong distribution class? Yung tube 1 natin, tube 2, tube 3, tube 4. After collection, yung pagkaka-red dito sa tube 1 is very distinct. Tapos sa mga sumunod, tawag dito, slowly fainting. Yun ba yung tamang term? Basta yun class. Okay, slowly nag-fade na yung kulay ng pagka-red ng sample natin. However, if the distribution here, yung sa test tube natin is even distribution, it is possible of hemorrhage. Okay? Now, in terms of clot formation or the presence of clot, a traumatic tap sample, okay, or rather a bloody sample due to traumatic tap, merong clot na mamumuo dyan. Okay? Positive for clot formation. Kasi bakit class? Okay? Okay? A traumatic tap sample, magkakaroon ng clot formation yan due to the action of fibrinogen. Okay? Due to the presence of fibrin. Ang tanong, meron bang fibrinogen sa meninges natin or rather sa CNS or the CSF sample itself? Wala. Kasi masyadong malalaki ang mga yan. Kaya, wala kang makikitang fibrinogen sa CNS natin. Okay? Now, for true hemorrhage naman, dapat wala kang makitang clot. Okay, now in terms of xanthochromia, xanthochromia class, later on we will be describing what is xanthochromia. Okay, yung xanthochromia na sinasabi nating class, it is only present in cases of true hemorrhage. Okay, xanthochromia. Dito wala. And then in terms of microscopic finding, okay, sa hemorrhage nating class, kung gusto mo talagang malaman na true hemorrhage yan, you should be able to find what we call your erythrophage. Okay? Erythrophages. Your erythrophage class, it is actually a type of macrophage that has ingested RBC. Yang macrophage na yan, merong RBC sa loob niya, indicating of a previous hemorrhage. Okay?